Good morning, students. I hope you all are good and welcome to today's session. And today's session is going to be on the chapter Principles of Inheritance and Variation of Class 12th Biology. So, as given in the description, we are going to start somewhere in the middle of the chapter because the other concepts have been covered. And the concept that I'm going to start with is going to be on the chromosomal basis of inheritance, and we'll complete the concepts all the way up to the sex determination in different organisms. So I just want a feedback. If you are able to hear my voice, please give a reply. Say yes or no so that I can continue. Good morning students. I hope you all can hear my voice. If yes, please do give a reply so that we can continue. Is the audio good? Okay, great. Then let us start. Okay. So in this chapter, we all understand the molecular basis of inheritance. So you must be aware of what inheritance means and what variations means. Now, to start with, let me remind you all what loci or locus means. So when we talk about locus, there is a lot of confusion whether it is present in the chromosome, whether it is part of the DNA, whether it is part of the gene. So you have to understand that locus is nothing but it's a physical location on a particular chromosome. So it is a physical location of a gene on a chromosome. For example, we have a gene in this picture that is going to control the eye color. So that is located at a particular location, particular place in the chromosome, and that is the locus for that particular gene, the eye color gene. Similarly, we have number of other genes. For example, we have another gene called the hair color gene, and this one is again present at a different location on the same gene, same chromosome. Now, the locus or the loci for different genes might be located on different chromosomes. But when we talk about alleles are nothing but gene variants or the genetic variants that is going to regulate one particular trait. So one gene regulates one character, and the gene variants, that is the allelic forms of that, are going to control or regulate the different traits of that. So in typical, what we have learned is each of the gene have a pair of allele. Although there are cases in which you will get more than one, more than two alleles, that is in case of multiple alleles. So as you can see here, this is a homologous pair of chromosome, that is you're going to have one copy from the paternal and one copy from the maternal, that is one chromosome from the father side, one chromosome from the mother side. So here you can see the loci. So this is a locus for what? For the coat, right? For locus for the coat in particular organism, coat color. So here you have one allele and here you have another allele. They are located in two different chromosomes, that is two different physical chromosomes, and this particular alleles are present at the same exact loci when we talk about the physical position on the chromosome. Now similarly, when we talk about the alleles, we all know there are dominant alleles and there are recessive alleles. So each of the allele are going to be located on two different chromosomes, that is one on the maternal, one on the paternal. So based on that, we can have homozygous for the dominant allele, we can have homozygous for the recessive allele, as you can see, small a, small a. Similarly, we can have heterozygous, that is we are having a dominant allele and we are having a recessive allele. So always remember, the alleles can exist as homozygous dominant, homozygous recessive and heterozygous. Now again, just a quick revision of the chromosomal theory of inheritance. Now till now, why this particular concept is important? So till now what scientists are unaware is what actually chromosomes are, because people have not found out yet. They are not knowing about what DNA or what genes are. So there are certain speculation on how it could be, and there was this scientist Nageli, and he started studying the chromosomal, the color bodied, and it was visualized under the microscope by staining, and thereby the name the chromosomes. And he studied all of these during the cell division, and he found that these colored bodies are going to double and they divide just before each of the cell division. So now there is going to be a cell division, there are going to be karyokinesis or the division of the chromosome or the DNA content or the genetic content here. And you can see here, these are some of the microscopic images of this particular process of the cell division. Now, the chromosomal behavior was studied extensively by Sutton and Boveri, and what they found is the chromosomal behavior is parallel to the behavior of the genes. Now, genes means we are talking about the factors as studied by Mendel, and these particular chromosomes also they behaved similar to those of the Mendel's factors, and they started explaining the Sutton and Boveri. They explained the Mendel's laws of inheritance using the chromosomal movement. I hope you are aware of the meiosis process and the formation of germ cell. As you can see here, there are in this picture you can see 4 different chromosomes, and these 4 different chromosomes, they are going to duplicate, and during the meiosis 1, in the anaphase they will start separating. The homologous chromosome will start separating, and at the end in the germ cell you get 1 copy each of the chromosome, right? But here what you see is, here the yellow one is actually getting segregated with the red chromosome, and the orange is segregating using the green chromosome. But again, when we talk about another condition, you can see here there is a long orange chromosome and short green chromosome, and again you have the long yellow chromosome and short red chromosome. Now the point of showing you this particular image is the independent assortment of the chromosomes. So the chromosomes here, they can independently assort, they can form the combinations in the germ cells independent of the partners or of the other chromosomes. That means the long orange chromosome can go with green, or the long orange chromosome can go with, as you can see here, red. Similarly, the long yellow chromosome can go with red, or it can go with green. So there is no linkage here, there is no fixed situation that a particular chromosome is going to go with another particular chromosome. So what is the conclusion? That the conclusion is that the chromosomes, like Mendel genes, come in matched pairs in an organism, that is homologous pairs in an organism. And for both the genes and the chromosomes, one member of the pair come from the mother and another from the father. So just like alleles are going to occur in pair, capital A, small a, that is one from mother and one from father. Similarly, the chromosomes, that is the homologous pair of chromosomes, are also present, in which you're going to get one set of chromosome, that is one chromosome for each pair of chromosomes, one from mother and one from father. The mother is going to be referred to as the maternal copy, and the the chromosome received from the father's side is going to be referred to as the paternal chromosome. Similarly, the second observations here, here the members of the homologous pair segregate in meiosis in such a way that each of the sperm or egg are going to receive only one copy of the member. So this is just like the law of segregation, right? So this process mirrors the law of segregation of the alleles into gametes in case of Mendel's law of segregation. So just like alleles, we had capital A, small a segregating into 2 different gametes, each gamete containing either the dominant form of the allele or the recessive form of the allele, or if it is in a homozygous condition, each of the gamete is going to receive only one copy of the allele. So similarly, the chromosomes also behave. So we have a pair of chromosome, and when they undergo the gamete formation, undergo the meiosis, at the end, finally, each of the gametes are going to contain only one copy of the chromosome and not a pair of the chromosome. Similarly, when we talk about the next law of independent assortment. So members of the different chromosome pairs are sorted into gametes independently of one another in the meiosis, just like alleles of different genes in Mendel's law of independent assortment. So as you can see here, we are having this heterozygous condition, capital A, capital B, small b. So here the capital A can go with capital B. The capital A can also go with small b. Similarly, the small a can go with capital B and the small a can go with small b. So here there is no fixed situation scenario that your capital A always go with capital B or small a will go always with small b. So there is no pre-decided scenario. So here capital A can go with capital B, capital A can go also with the small b, that is the recessive allele. So this is the same way that the chromosome behave. That is, as you can see here, the chromosomes, that is the long chromosome A, capital A, and here you have the small a. Similarly, the long chromosome capital B and small b. So here you can see, here you are going to have capital A, capital B, capital A, capital B, but here also you have small b. But in another situation, again, it is all about the meiosis. So in another situation, we can have capital A, small b, and capital A, small b, small b is capital B, small a, capital B. So there is a combination, there is a chance that the chromosome, one chromosome that is represented by this capital A can go with the capital B, or it can go with the capital A can go with also the small b. Similarly, the small a can go with capital B, or the small a can go with small b. So the behavior of the chromosomes and the alleles are similar, and based on this they provided the chromosomal theory of inheritance. But again, as always, if there is no experimental verification, people are not going to believe. So the experimental verification for this chromosomal theory of inheritance came from the work of Thomas Hunt Morgan and his colleagues. And he worked on the fruit fly, that is Drosophila melanogaster. Now the question is, why did he choose the Drosophila melanogaster or the fruit fly? Similarly, Mendel chose the pea plant. There were certain reasons for that. Here also there are few reasons for it, and it is listed here. Now one of the primary reason is these organisms, these fruit flies, could be grown on simple synthetic medium in the laboratory. There is no extensive preparation, there is no complex method of growing them. Simple synthetic medium is good enough for them to be cultured in the laboratory. Second, just like Mendel's pea plant, here also this fruit fly, they could complete their life cycle in a short period of time, and that is only about 2 weeks of time. So, and if this lifespan is small, if the life cycle is small, the amount of studies that can be done generation after generation is going to be much faster. The third point is, a single mating could produce a large number of progeny flies. Now progeny flies. So here in a single mating, single reproduction, single generation, the number of progeny that we get is going to be much better, and it is easier for us to make the studies. And another reason was a clear differentiation of the sexes, that is, the male and female flies could be easily distinguished. We'll see why it is very important to identify the male and female in this case. And one of the last reasons is it had many types of hereditary variation that can be seen with a low power microscope. And when we talk about hereditary variation, for example, the difference in the wing pattern, the difference in the body color, the difference in the color of the eye. So these are easily identifiable under a simple microscope. And these were the reasons why Morgan chose the Drosophila. So what is this experiment about? So Morgan performed again dihybrid cross in Drosophila to study the genes that are sex-linked. He tried to understand whether there is something called sex-linked inheritance. So what Morgan did, Morgan hybridized yellow-bodied white-eyed females to the brown-bodied red-eyed males, and again intercrossed their F1 progeny, just like Mendel's experiment F1 and F2. So here the parents were yellow-bodied white-eyed females to brown-bodied red-eyed males. And till now, based on our understanding, we will guess that it is based on the Morgan, Mendel's experiment. It is, that they will be getting this regular ratio of 9 is to 3 is to 3 is to 1, and this is the case only if the genes are segregating independent of each other, right? Till now we have seen independent assortment of genes, right, or alleles. So if they are following the Mendelian inheritance, they will give us the regular 9 is to 3 is to 3 is to 1 ratio of the F2 generation. But the thing is, what Morgan found, that the genes did not segregate independently, right? They did not segregate independently of each other, and here the F2 deviated very significantly from the regular ratio of 9 to 3 to 3 to 1 of a dihybrid Mendelian cross. So what are the reasons for that? So they knew that the genes they were trying to study is actually located on the X chromosome. Okay, and what is the observation then? So when two genes in a dihybrid cross are located on the same chromosome, right? We are talking about X chromosome. There are two genes on the X chromosome. So the proportion of parental gene combinations, right? Whatever combination of gene we had in the parent were likely to be much higher than the non-parental type in the progeny. So when we talk about the F2 generation, we find more of the parental type of the gene combination rather than a new combinations, and that is the reason why we are deviating from the 9 is to 3 to 3 to 1 ratio. Now why is this happening? So the justification that came around by Morgan was there is physical association or linkage. Okay, remember this word, very important. So there is physical association or linkage of the two genes. So what is linkage? Linkage is the physical association of the genes on a chromosome. We'll see that in a minute. And he also used a term called recombination. Linkage was the term introduced by Morgan, and also the recombination. So recombination he used to describe the generation of non-parental combination of traits. So if it is something similar to that of the parent, then it is non-recombinance. There is no recombination. Again, why am I talking about this? See, if you are aware of the meiosis, you know that in the meiosis 1 prophase 1 there is going to be recombination of the chromosomes. There is that meiosis, there are meiosis 1 of prophase 1, the different phases, there is going to be exchange of genetic material. Now if that is happening, if that exchange is going to be there and the genes are present in two different parts of the chromosome, then we will get this recombinance. That is something which was not present on the parents. That is something new. So the recombination describes the generation of non-parental combination of the traits. So let me go back and understand a little bit of basics why this is happening. So again, all of these images that I'm going to show you in the next 4 or 5 slides are actually from Khan Academy. So anyway, so here the genes, if they're on different chromosomes, as you can see here, we're having gene A and gene B, and for each we know they are going to be present in the two different of the homologous chromosome of the same gene. So this is one homologous chromosome, this is another homologous chromosome. Now here the genes are present on two different chromosomes. So when the gametes are made, what happens? We get gametes of different combinations, each with 25%. That is 1 is to 1 is to 1 is to 1 of capital A, capital B, small a, small b, small a, capital B, capital A, small b. Now what if the genes are on the same chromosome, right? Now on the same homologous chromosome we have both gene A allele dominant recessive, gene B allele dominant and recessive. But here also when we are going to get the gametes are going to be capital A, capital B, small a, small b, small a, capital B, and capital A, small b. That is all are going to be in the 25% proportion, or 1 is to 1 is to 1 is to 1 ratio. Now why this happens? Right, try to understand this. We are having homologous pair of chromosome. So that is, we are having one copy from dad, that is from the father, and one copy from the mother. Now when DNA is going to replicate for meiosis, they are going to duplicate, right? And you're going to have the duplicated set of homologous chromosomes. Now you can see here, when you're going to have this duplicated set of homologous chromosomes and they segregate, we get these different combinations of chromosomes. That is capital B, small b, small a capital B, and small capital A small b, all in the 25% proportion. And the reason for that is the segregation of chromosome that is taking place in the meiosis 1 and meiosis 2. Now when we are going to have okay, so when you're going to have the two gene on the same chromosome and they are very close to each other, right? So here the chromosomes are going to be only one set of chromosome, and you're going to have two genes, and the two genes are located very close to each other. Now what happens is, here the gametes that are going to be made is going to be slightly different. That is, in most of the cases you're going to get a high chance of capital A, capital B, and a high chance of small a, small b. That is the parental combination. See, capital A and capital B, small a and small b. So this is called the parental combination, parental genes. Now a very low chance is there for the recombinants. There is a very low chance that this capital A will go with capital B and the capital B going with small b. The chances are very low, and this why these are called recombinants. The question is why? See, when the two genes are nearby in the same chromosome, right, the only crossover happening in this small region, right, in the small region, that only can produce the non-parental combination, that is heterozygous capital A small b or the small a capital B. So the recombinant chromosomes do form, but very rare. They're not very often formed, right? That is why, as you can see here, there is a 48% of the parental combination, 48% here, 48% here, and here it is only 2% for the recombinant type. And the reason for this is going to be, there is a physical linkage, physical relationship between these two chromosomes that are nearby. So what is the solution? What is the summary of all of this? When the genes are grouped on the same chromosome, right, they are present nearby, the genes can be very tightly linked, right? So when the genes are on the same chromosome, the genes can be tightly linked, and when they're tightly linked, they're going to exhibit a strong linkage, and that is going to exhibit a very low recombination. Okay, but when the chromosomes are on the same chromosome, right, and when the genes are on the same chromosome but are loosely linked, in that case the linkage is weak, right, weak linkage, but in that case you are going to get a higher rate of recombinations. So just to give you a summary from your textbook, as you can see here, in the yellow and white case, the genes are nearby, right? So here the genes are nearby. So what do you get? You get in the F2 a whooping 98.7% of parental type, but only a 1.3% of recombinant type of the alleles here, of the genes here. But when the genes are far apart, they are on the same chromosome, W and M, wide and miniature. So here what happens? The genes are located far apart, but here they were very close. So when the genes are far apart, what happens? There is going to be a weak linkage, and when you have weak linkage, there is going to be high rate of recombination. So here what you get a recombinant of considerably much higher, that is if you compare 1.3, but here it is 37.2% of recombinants, and a parental type of only 62.8%, reduced from the 98.7%, right? So here what is happening? The recombinants in the progeny, in the F2, it depends on the linkage. If the genes on the same chromosomes are nearby, the linkage is going to be strong, thereby the recombinant types, percentage of recombination is going to be less. But if the chromosomes are far apart on the same chromosome, in that case the linkage is going to be weak, thereby a higher rate of recombination. I hope I am clear with that. Now again, this is the same explanation, right, using a image from, this is from Encyclopedia Britannica. As you can see here, same white-eyed female, red-eyed females male were crossed, and you get the outcome. It is again going to deviate from that of the regular or 9:3:3 3:1 ratio. You can have a look at this also, but again this is not there in the textbook, so you can just further understand, you can follow it. Now, based on all of these studies, the student of Morgan, that is Alfred Sturtevant, he used the information from the study, that is the frequency of recombination. क्या डेटा लिए थे? Frequency of recombinations between the gene pairs on the same chromosome, and he measured the distance between gene, and he created something called the genetic map. So what is genetic map? Genetic map is like it's the information in the entire chromosome where we are having which chromosome. So this genetic map for the first time was developed by Sturtevant, student of Morgan, and he used the frequency recombination to develop this particular genetic map. Now we're coming to the next part, that is polygenic inheritance. I hope I am getting clear, the students. Okay, let me check a little bit. Yes, okay. I'm not able to see the chat and the the slides together, so I'm going to come back, and I'm actually checking the chats now. So yes, very nice. Linkage recombination, yes, linkage recombination are going to be inversely proportional. Stronger the linkage, weaker will be the recombination frequency, and stronger the weaker the linkage, higher will be the recombination frequency. Yes, indirectly proportional to the linkage. Yes. Okay, good. Anyways, so I hope you are clear with that. If not, anyway, we can get back to the session, doubt clear session towards the end, so that I can focus on that part. So right now, let us go and talk about the next concept, that is polygenic inheritance. Okay, good. Thank you all. So till now, what you have seen is inheritance of one gene or two gene, right? And we have learned that one gene is going to control one trait. One gene regulates one character, and that character is character or traits are going to regulate by those alleles, the two alleles part of that single gene. But we also have learned that till now there are contrasting pair of characters. That is, we are going to have dominant traits, and we are going to have the recessive traits. And we till now did not talk about any intermediate range of traits. But again, in nature, everything is not going to follow into that, right? So there are certain characters that are not going to follow the dominant trait or the recessive trait, but instead they show an range of traits. For example, the eye color, the skin tone, or the skin color. There is nothing called very bright and very dark, but there are intermediate colors also. Now, how do you explain these things? How do you explain the inheritance of these traits? So we are going to talk about polygenic inheritance and explain this exact same point. So here, traits, when it is controlled by more than two genes, right? Two genes or more than two genes, that is called polygenic inheritance. Many gene, more than two gene or more than two gene, that is called polygenic inheritance. And it tries to explain the traits that are not exactly the contrasting pairs, but it's a range of trait. To give you the example of the skin colors, skin pigmentation, right? We all know there's a range of it. And here what happens is there are three different genes that are going to regulate the skin color, the skin pigmentation. What are those? We are going to have A, B, C gene. Simple, A, B, C gene. Now, both of these, all these three genes are going to have the dominant form as well as the recessive form. So what happens here is the nature of the skin pigmentation depends on the number of dominant allele and the number of recessive allele. So when you're going to have a completely recessive condition, that is A, B, C, all the three genes are in complete recessive condition. In that case, we get the very light skin color. The skin pigmentation is almost nil. See, again, we know that the skin pigmentation is dependent on the amount of melanin production. So here, the skin pigmentation is very, very light. But when you're going to have all the three gene, all the six alleles dominant, it is going to be the darkest skin pigmentation. So here you can see there is this particular the inheritance pattern. So if you are having small a, small b, small c representing the very light condition, that is zero dominant, 0D, right? Here you're going to have the zero dominant, but here you are going to have the 6 dominant alleles. So you are going to have the very dark condition. Now in the F1 generation, it is all intermediate. So here you are going to have only 3 are dominant and 3 are recessive. Now in the sperm and egg, again you can have any 3 of these. So you can see in this Punnett square, so we can have all the way from 0 dominant all the way to 1, 2, 3, 4, 5, 6 dominant. And the skin pigmentation here, remember, depends on the number of dominant alleles. So here, in other words, we can say that the phenotype is going to be reflected by the contribution of each of the allele. So here the effect, the phenotypic expression of the alleles are additive. That is, if one of the allele is dominant, less phenotypic expression. If 2, slightly more, 3 slightly more, and at the 6 it is a maximum. So here the effect, the phenotypic expression of the alleles are going to be additive. That is what you have to remember when we talk about polygenic inheritance. And again, you can see here in a population there are going to be all these traits, but again it is going to be a bell-shaped curve with the middle, that is going to be your intermediate form, is going to be the most common in the population. So here, as you can see here, there are going to be different genes regulating a single character, and that time we call it a polygenic inheritance. And as we said, depending on the number of dominant traits, right, you can see here number of dominant traits 0, 1, 2, 3, 4, 5, 6, you can have a skin pigmentation from very light to medium to very dark, right? So the next one is going to be pleiotropic, right? Pleiotropy is different than polygenic inheritance. So till now what you have seen is a single gene regulates a single phenotypic expression. But when there is a single gene and it regulates more than one phenotypic expression, that situation is called pleiotropy, and the inheritance is called pleiotropic inheritance. As you can see here, you have a single gene, right? And the single gene is going to regulate various traits. It can be 1, 2, or maybe 3. So when you are going to have a single gene regulating more than one trait, multiple phenotypic expression, it is called pleiotropy, and the gene is called pleiotropic gene, and the inheritance pattern is called pleiotropic inheritance. One of the examples that you have to understand based on your textbook is going to be on the disease PKU, or phenylketonuria. You're going to learn about it in detail at a later point of time when we talk about the disorders or diseases. So what happens in here is the mutation in the gene that codes for the enzyme phenylalanine hydroxylase. So there is an enzyme called phenylalanine hydroxylase, and a single mutation in this that leads to the PKU, or phenylketonuria. So here the expression of the gene, so because of the single mutation in a single gene, a change in a single gene, it leads to more than one phenotypic expression. So what are those? That is mental retardation, right? Mental retardation is one expression, right? The second one is reduction in the hair and skin pigmentation. So as you can see here, there is reduction in the hair and skin pigmentation as well as the mental retardation, and both of these are going to be two different phenotypic expression, and they are going to be regulated by a single gene. So we are seeing this because there is a mutation in a single gene, not two different genes. Ideally these two are two different traits, so they should be regulated by two different genes, but no, here it is a single gene and that regulates both of these, and that is pleiotropy at the pleiotropic gene. Again, just to give you a summary, polygenic inheritance means you have more than one gene regulating a single character or single trait, whereas in pleiotropy you are going to have single gene regulating more than one phenotypic expression. It is opposite. Both of these are opposite. Do not confuse with both of these traits. So we are now coming to the sex determination. How the sex of an organism, male and female, is determined at a genetic level. So what happened? How did people learn about this? So scientists were actually looking at the behavior of the the chromosome, certain nuclear structures of various insects. And one of the prominent name that we have to understand is Henking. In the year 1981, he was actually tracing specific nuclear structure. At that time, people did not know, so he also was unaware of what it was, but he was tracing a specific nuclear structure throughout the spermatogenesis. You have known that spermatogenesis is the process of the sperm formation, right? Gamete formation in case of males, in case of few insects. And what he found that in those insects, 50% of the sperm received this specific nuclear structure that he was tracking, but the remaining 50% did not receive it, right? Only 50% of the sperm receives, but the remaining 50% did not. So what could it be? That, you know, it is partial, it is going to somewhere, it is not going to some other places. And because he didn't know, he didn't know what it was, he gave the name X body, and he could not explain the significance. But later on, with the advancement in studies of the other scientists, you know, they found that X body, that unknown specific nuclear structure that Henking was tracking, was nothing but a chromosome, and they gave the name X chromosome. And later, after a number of other studies, they have found that this X chromosome, right? X chromosome is going to determine the sex in many of the organisms. Again, there are different ways, different types of sex determination that we're going to talk about today. So the first one, let us talk about insects only. For example, in case of grasshopper, that we have, XO type of sex determination. Right, O is nothing, it's not a chromosome, but it simply refers to the absence of a chromosome in that pair of chromosome. So it is exhibited by a large number of insects. So here, all of the eggs, they contain an extra chromosome called the X chromosome. But when you talk about the sperm, 50% have it, 50% do not have it. So how it is, represented? So we have all the autosome, right? Along with that, we have XO. Only X chromosome is there, right? There is only a single X chromosome, and they are the males. Keep it in mind. XO represents the males. But in case of female, this is a pair of X chromosomes. So you have XX, and here there is no XO condition. Here it is XX. So if you are having autosome with a pair of X chromosome, it is the female. Now, when the gamete is going to be formed, the sperm contains all the a set of autosomes along with a single X chromosome, or it might contain only all the autosomes and no sex chromosome, right? No X chromosome, it is only the autosomes. One chromosome is less in here. Similarly, in case of female, there is only one type that is going to be formed, and that is all the autosome with a single X chromosome. And you understand by this process that when this particular sperm with AX configuration fuses with this particular ova containing AX, you get XX, that is going to be female. Similarly, when you're going to have the XX, when these two are fusing, again there is a female. But when you're going to have this particular sperm that is only with the autosome fusing with one of the ova, you're going to get the male, as it is going to have all the autosomes only with one sex chromosome, one X chromosome, and no pair condition. So only you are going to have one X chromosome, all autosome with X chromosome. So XO. So this is the this is the XO type of sex determination we see in case of grasshoppers. Now, when we talk about the humans, right, and some of the other organisms, we see the XY type of sex determination. So here, the male and the female have the same number of chromosomes, right? While in the previous case, as you saw, there is a difference in the number of chromosomes in some of the gametes, right? Here, there is no difference. Both the male as well as the female have the same number of chromosomes. The difference here is the males have autosomes with XY configuration of the sex chromosome, right? You have a, I'm sorry, this is but typed wrong. This is going to be, right, XX for the female, but in case of male, you're going to have A and XY configuration. That is, you're going to have a larger X chromosome and a smaller Y chromosome. Now, you see this condition in case of human beings and also in case of the Drosophila melanogaster, the fruit flies. Now, in both of these cases, what you have seen is there are organisms, there are a male or female that is going to produce either the same type of chromosome of the sex chromosome or different type. And both based on that, we are having two terms. One is called heterogamety and the other is called homogamety. Hetero, different type of that gametes. So production of sex chromosome that differ in morphology, resulting in the formation of two different kind of chromosome, that is called heterogamety. And both in XO and XY type, it is the males that are heterogametes. So it is called male heterogamy. Now, in case of the female, in both of these XO and XX type, the only one type of gametes are formed. So it is called homogamety. So it is called female homogamety. Production of sex chromosome that do not differ in morphology, resulting in the formation of only one kind of gamete, that is called homogametes. So in case of XO and XY, only the males are heterogametes. But when we talk about birds, right, that is slightly different. In case of birds, what happened? The female is heterogametic, not the male. And here there is no XY, but here we have the Z and W chromosomes as the sex chromosome. So in case of female, we are going to have, as if the female is heterogametic, we are going to have all the autosomes along with ZW configuration of the sex chromosome. And the males are going to have autosomes with a pair of Z chromosome. So the sperm contains a set of the autosomes with a single Z chromosome, and the ova can contain autosomes with either Z or W chromosome. And based on that, when they combine, when you are going to have that is your autosomes with a pair of Z chromosome, you are going to have the male, and when you are going to have the autosome again with a ZW configuration of the sex chromosome, you are going to have the female. So this is a ZW type of sex determination. Here it is opposite. Here in the previous two cases, the male were heterogametic, but here the female is heterogametic. This is what you have to keep in mind. Many times there is confusion on which one is male heterogametic, which is female heterogametic. Now, how does the sex determined in the human beings? Many of you might be aware, but again, just a brief recap. So we all know that diploid cells in humans are going to consisting of 23 pairs of chromosomes, that is 22 pairs of autosome and one pair of sex chromosome. And each of this pair, when you talk about pair, one is paternal, one is maternal set of chromosomes. So the 22 pairs of chromosomes are nothing but the 44 chromosomes with XY in case of male and 44 chromosome and XX chromosome in case of female. Similarly, the sperm contains 22 autosomes with X or 22 autosomes with Y chromosome, but all the female gamete contain 22 chromosomes with X chromosome only. So when the sperm containing X chromosome fuses with the sperm containing X chromosome, it gives rise to a female, whereas when a sperm containing Y chromosome fuses the sperm containing X chromosome, egg containing X chromosome, it is going to give rise to a female. So till now what you have seen is, in all of these cases, there is a 50% probability of the child being male and a 50% probability of the child being female. And in case of humans, the chances, the probability of male or female is dependent on the male partner, as the sex of the child is dependent on what kind of sperm cells are going to fuse the female, whether it is X or it is Y. Now, till now what you have seen is a genetic basis. We are talking about X chromosome and Y chromosome, X chromosome or no chromosome or ZZW chromosome. And we said till now about the sex chromosome. But in case of honeybee, the sex determination is completely different. Here it is based on the number of chromosomes. Here it is called the haplodiploid sex determination, right? Why it is called? Let us see. Now, the female here is going to be having 32 chromosomes. The first thing you have to keep in mind, the female is going to have 32 chromosomes, whereas the male is going to have only 16 chromosomes, right? Now, the female give rise to the gametes, and the gametes are going to have 16 chromosomes each, right? All the gametes are going to have 16 chromosomes, half of that haploid. But the process here involved is meiosis. The regular process, no difference. But you have to keep in mind that in case of male, in case of the honeybee, the male give rise to the gamete by mitosis and not meiosis. So here the male gametes are formed by mitosis and not the regular process of meiosis. Now, when the male gamete containing 16 chromosome fuses with the female gamete containing 16 chromosome, it becomes a 32 chromosomal structure, and that organism develops into the female. Right? As we said, if 32 chromosomes, then it is going to be a female organism. Now, what about the male? So here, very interesting, the 16 chromosome gamete from the female, right, that is not going to fuse with the male, it can develop into a 16 chromosome organism by the process of parthenogenesis, right, and they develop into male. So in simple term, the male organisms are produced here by parthenogenesis and not the regular process of fertilization. So in simple term, we can say that the male bees do not have father. You can see here the male bee, they have father? No, they don't have father. They have only the mother. So the male bee do not have father. And now the question is, these male, do they have son? No, they have only the daughters. So the male bee do not have father nor the sons. Now, can they have grandfather? Yes. So the male bee can have grandfather and can have the grandson. Now, why this statement is important? Many times they ask why this statement. No, why the male bees do not have father and no son, but they can have grandfather and grandson. Explain. So you have to basically draw this particular inheritance pattern and explain why this happens. What is parthenogenesis? How the parthenogenesis is playing the role here. So here the female bee can be a queen or the worker, whereas the male is going to be always produced by the parthenogenesis and they are going to be always the drone. So that is going to be the sex determination in the honeybee. And with this we are coming to a close of this particular concepts of the sex determination in honeybee. So now, dear students, if you have any doubts, please do ask. I'll take your questions now. Yeah, Mansi, good morning. Okay, I'm going to take few questions here. Let me see. Completing linkage and complete linkage. Yeah, Jyoti, will be talking in detail about that also at a later point of time. Hair color is also hair color could be a character, no, different hair colors, right? Don't always think about the Indians hair color, but again, if you go to other countries, there are different kind of hair colors. That is also a character, and they are not the same, right? There are albino condition where the hair color is always going to be white. There's a kid even in my locality who is having a completely white color hair, and she is an Indian, right? That girl is an Indian, small kid. So that is why the hair color is also could be a melanin content depends. Yeah, Panigrahi, melanin content is going to be dependent on that. Okay, what is the role of A in the chromosome? Dear Preeti, A is nothing but it is depending the chromosomes of the autosomes. Biswanath has said. So A are nothing but the autosomes, all the autosomes, 23 autosomes. Okay. Yes, they decides character other than sex. Yeah. Yeah, Mansi. Good morning. Yeah. Okay, dear students, any other doubts? Please ask. If not, we can end the stream today. Again, I am going to have a meeting today. I have to attend to that. A training session is there at 11. So if the doubts are clear, we can end the session and we can continue the next session. Okay, I think I'm just going to ask in the chat, should we, okay, let me check here. Should we close? Which is I'm having it at 11 o'clock, so that time I have to go. Say training by CBSE RO. Thank you all for joining. Okay, yeah. Just a minute in the WhatsApp group. Just be there. Okay, and I'm going to end the stream here. Yeah, thank you all.